Hello everyone, welcome back to your classes on overview and integration of cellular metabolism. We are continuing with amino acid metabolism. This is the last class of amino acid metabolism. We will be discussing amino acid metabolism when we move to integration section and inborn of metabolism. Today's topic is metabolism of histidine, proline, arginine and lysine. We have a lot to cover and these are the concepts that we will be discussing today. We will be discussing the important product from each of these amino acid. We will be discussing the metabolism of each of these amino acid. For example, for arginine we will be discussing nitric oxide, for histidine we will be discussing histamine. We will be discussing how these all amino acids are connected, what is the interrelationship among them and their integrated metabolic fate. So, let us start right away. Histidine, alright. Histidine, why it is special? If we look at the chemistry, it has got an imidazole ring, this side chain, this pentagon, right, 5 carbon chain which has got 2 nitrogen, imidazole ring. It is a basic amino acid by its chemical nature and it is a semi-essential. I told you the amino acids that are not fully required in diet, but their function can be essential when there is growth development, right. So, histidine falls in that category, alright. So, it is a semi-essential basic amino acid, right. Now, one thing that histidine is known for is its buffering action. This will not be discussed in this class. This is a concept of acids and bases. You all need to know what is the pKa, that is the dissociation constant of the amino acid, how they dissociate in an acidic or basic medium. The pKa of histidine is actually 6.1, which imparts to its maximum buffering action because it is very near to the physiological pH, and histidine, as it is present in hemoglobin, we will be seeing when we discuss heme synthesis in the next class, it helps in this physiological activity, right. So, these are the few things about histidine, the chemistry. So, regarding the metabolism of histidine, what we need to know is how histidine is degraded mainly, right. So, histidine by the action of histidase is converted to urocanate, or in some cases it is also known as urocanic acid, which upon action by urocanase enzyme is converted to 4-imidazolone 5-propionate, right, which undergoes a hydrolase enzyme action, right. Hydrolase, what hydrolase do? They breaking down of the molecule by incorporating water molecule, right. On action of an hydrolase, it forms formiminoglutamate or N-formiminoglutamate, the most important intermediate of histidine metabolism, which is also abbreviated as FIGLU, F-I-G-L-U, formiminoglutamate, right. This formiminoglutamate actually can be converted to glutamate involving a 1-carbon metabolism, right. Tetrahydrofolate forms N5 formiminotetrahydrofolate. I told you when we are discussing 1-carbon metabolism that this belongs to a 1-carbon pool. 1-carbon pool is basically those molecules which participate in 1-carbon reaction, and thus histidine, actually histidine metabolism also contributes to the 1-carbon pool by formation of N5 formiminotetrahydrofolate, and this glutamate can be converted to alpha ketoglutarate by transamination, right. So, and that alpha ketoglutarate, which is an intermediate of TC cycle, can always be converted back to glucose by reversal of glycolysis and gluconeogenesis. So, histidine is glucogenic, there is no doubt about it, alright. As I discussed, histidine also contributes to the 1-carbon pool by formation of formiminotetrahydrofolate, but one important thing to remember is because of this reaction, that is histidine is converted to glutamate and the intermediate is formiminoglutamate, right, and conversion of formiminoglutamate to glutamate needs folic acid, right. So, whenever there is a deficiency of folic acid, FIGLU will be accumulated and it will be excreted in urine. So, actually FIGLU excretion is a marker of folic acid deficiency, a very important applied question for you. So, let us know how this FIGLU excretion excretion test it actually performed the exact procedure. The what is the amount of histidine, this is also known as histidine loading dose. So, histidine is loaded, I mean excess dose is given 3 times at 4 hourly interval, 5 grams of each. Okay, this is the dose. What happened? Urine is collected 24 hours after the initial dose. So, we will measure the total amount of figlu that is excreted in 24 hours of collected urine, and generally it is less than 30 milligram per 24 hour, and this is raised in folate deficiency. Alright. So, this is the cutoff value beyond which it will deal with folate deficiency. Now, this test is not done routinely. Why? Because we can directly measure folic acid very accurately nowadays with the help of enzyme linked immunosorbent assay or ELISA. So, know this figlu excretion test is important, it was important, but nowadays it is mostly historically important because we have got ELISA in almost all, even peripheral, some peripheral centers also, right. So, if we got ELISA, figlu excretion test is historically important, right, but still for metabolic point of view it is very important. So, as I told you, we will discuss all the important products from histidine, right, and one of them is histamine, right. Regarding any competitive exam or any exam, this is the reaction or this is the product that you need to remember, right. The intermediates of the previous reaction, the exact intermediates, what was the sequence of reaction may not be so important for you to remember, that is the histidine degrading pathway, right, but this is important. Figlu, that step of course it is important, right, but here we should know that histidine by the action of a decarboxylase enzyme, decarboxylase means it loses one COO carboxyl group in the form of carbon dioxide and it forms histamine, right, and this is very important. In fact, it is so important that question often comes regarding histamine metabolism or degradation of histamine compared to degradation of its parent amino acid histidine. So, let us discuss while we are discussing this, histamine, just like catecholamine when we were studying tyrosine, right, you were taught how the catecholamines like adrenaline and noradrenaline were being synthesized, right, and catecholamine degradation is also equally important to know. If you recall, catecholamines were degraded by 2 enzymes, MAO monoamine oxidase and COMT catechol orthomethyl transferase. The mechanism is almost similar over here. Here since it is a diamine, so diamine oxidase and histamine N methyl transferase, simple. So, methyl transfer and amine oxidation. So, it forms, if it is acted upon by diamine oxidase, it forms imidazole acetaldehyde, and if it is undergoing a methyl transfer, it forms N methyl histamine. Now, know this, both of these products can actually feedbackly inhibit their formation, right. So, this is a feedback loop where the action of DAO and HNMT can be inhibited by imidazole acetaldehyde and HN methyl transferase respectively, right. So, what happens next? By the action of aldehyde dehydrogenase, both of them can be converted to their final excretory form, that is imidazole acetate or N methyl imidazole acetate, right, fine. So, these are the enzymes, and even on this N methyl histamine DAO diamine oxidase can act to form N methyl imidazole acetaldehyde. So, these are the final forms in which histamine is finally converted to when the action of histamine is over. So, these are the enzymes. So, histidine decarboxylase, diamine oxidase, methyl transferase. The fourth is the feedback loop that is being shown, again monoamine oxidase B, very important. MAOA was acting in catecholamines, on catecholamines, this is MAOB isoenzyme, right, and then again this reaction is catalyzed by diamine oxidase. So, why histamine is so important? Again, discussion about histamine, detailed discussion about histamine is out of the purview of this metabolism lesson, but here is an overview of histamine, what it can do. Most of you may already know that histamine is the mediator of all allergic reaction, all anaphylaxis, urticaria, pruritus, etc. are done by histamine. Histamine acts by multiple subclass of receptor H1, H2, H3 and H4, and one of the example, that is H2 receptor, helps in secretion of gastric acid. So we often take medicines like ranitidine, famotidine, right, Rantac, Famtac, those are H2 receptor blocker that will help in decreasing gastric acid secretion. So that is one example. Again, those antihistaminics are anti-allergics, how it is used. So all the reactions, that is pruritus, urticaria, hot flushes, etc. are there. Apart from skin, it is also acting on cardiovascular system, it is acting on nerves, it is acting on bone marrow, leukocyte, respiratory tract, uterus, gastric epithelium, everything, right. So you can actually form an endless list of what are the actions of histamine. So it is that much important. So you should actually divulge from the metabolic lessons and make a note of yourself, or make a point wise note what are the functions of histamine. It will be discussed more or later when you are studying pharmacology, because all these histamine receptor blockers are actually active pharmacological agents or medicines. Okay. There is another dimension of histidine metabolism, that is metabolism of dipeptides. This is not the normal way in which histidine is metabolized, this is the preferred way, histidase and urocanate pathway. Nevertheless, histidine is also involved in synthesis of compounds like anserine and carnosine, also 1-methyl-histidine, by action of multiple enzymes. For example, it is carnosine synthase which forms carnosine. Carnosine N-methyl transfers, again play of one carbon, it forms anserine. Anserinase degrades it into 1-methyl-histidine. Again, histidine can be converted to 1-methyl-histidine by a methyl transferase. One very important thing to know is you are seeing a role of an amino acid which we discussed as a variant of alanine when we were discussing alanine, that is beta alanine, right. So beta alanine plays a role in histidine dipeptide metabolism. So why do we need to know the role of anserine and carnosine? Well, they are very important antioxidants. Okay, free radical are tackled, or oxidative stress are tackled by these intermediates or these compounds, right, and they have been shown to reduce cognitive decline in research studies. Okay, so you should know that, at least you should know these are the products from histidine metabolism, even if you do not remember the entire pathway. So you can jolly well have an MCQ. All of these compounds are products of histidine metabolism, except you can get anserine, carnosine, urocanate and sarcosine, very close related name, carnosine and sarcosine. We read sarcosine in glycine and serine metabolism. So be very careful regarding your questions, and if you are mindful and remember your lesson well enough, it will be good to answer any questions. Okay. So what are the disorders of histidine metabolism? The first disorder that we need to know is histidinemia, autosomally recessive inheritance, absence of histidase enzyme. So normally what histidase does, it helps in conversion or breaking of histidine to urocanic acid. If that is not happening, naturally histidine will be diverted to alternate routes of metabolism, very similar to what was happening in phenylalanine metabolism. Over there phenylalanine was being converted to tyrosine. If phenylalanine hydroxylase was deficient, it was getting converted to phenyl acetate, phenyl lactate, phenyl pyruvate. Same thing or almost similar thing is happening over here. Histidine, if it is not acted upon by histidase, it is forming imidazole pyruvic acid, imidazole lactic acid and imidazole acetic acid, right. And all these intermediates, you are just required to remember the names, and these intermediates will lead to features of amino acid metabolism defect, disorders of amino acid metabolism, which is mental retardation and delayed speech development. All right. How we can prevent this? If we diagnose this disease, naturally we need to reduce the load of histidine in diet, right, and generally it may have some protective effect, right. But know this is the autosomal recessive disorder, it is not always expressed unless both the genes are recessive and homozygous. Okay. You can, if you have studied phenylketonuria well enough, you can easily draw the analogy to this. Okay. Next, urocanic aciduria. What is urocanic aciduria? Defect in the urocanase enzyme. So, urocanase was converting urocanate to 4-imidazolone 5-propionate. If this is deficient, all the previous intermediates will accumulate. So, it is very easy to remember, urocanic acid and histidine are excreted in urine, right. However, it is good compared to other diseases that clinical manifestations are minimal, okay. Only the high level of these compounds are found. Now, imidazole aminoaciduria. What is imidazole aminoaciduria? It is a defect in renal transport mechanism. So, these amino acids and its products, its dipeptides are excreted in large amount, that is histidine and 1-methyl histidine, right. We already read this reaction a while back. So, these intermediates will be excreted in urine, right. This is very important because it leads to problem in eye, cerebromacular degeneration and retinal degeneration, which may lead to blindness, right. And the defect in urinary transmission or renal defect is actually transmitted as a dominant trait, right. So, excess amino acids may be excreted in urine, that is present in majority, but the cerebromacular degeneration is actually recessive trait. So, it is good, it is beneficial in a way that even if the disease is present in most individual, the symptom, the dangerous symptom of blindness appears, is present very less, right. So, remember, imidazole aminoaciduria, selective receptor defect or selective transport mechanism defect in which histidine, that is this imidazole amino acid, is excreted in urine, right. So, after histidine we now focus on to proline, right. Proline is a non-essential glucogenic amino acid, right. Whenever we are discussing non-essential means we at least need to know how it is synthesized. Proline does not participate in transamination reaction. What was the another amino acid that was not participating in transamination, right. This is your homework. I hope you have recalled your lessons well enough and you should be able to answer it, right. So, this is the synthesis of proline, okay. So, you can see proline is first acted upon by an enzyme delta 1 pyrroline 5 carboxylate synthetase. This is a big intermediate, it is abbreviated as P5CS, and it is in the similar step that this enzyme or this compound will be reduced as well, and this enzyme synthetase by the action of ATP, reductase and dehydrogenase are all acting, but the only important thing for you to remember in this whole synthetic pathway is the source of synthesis is glutamate and ornithine, most important source being glutamate, and since ATP is used, this is a synthetase enzyme, right. A very important intermediate of glutamate, semialdehyde, is formed, right, and it undergoes a spontaneous cyclization to form proline. Now, you may think that this discussion is not in depth, right. Well, it is on purpose because when we will be studying degradation of proline, you will see this is the exact reversal of this pathway, right. So, ornithine is a minor source of proline, glutamate is the most important source, because when proline is degraded, it has a tendency to form glutamate and also ornithine, right. So, as I told you, this is the exact reversal during degradation of proline. So, what happens? The exact reverse steps are happening. So, first of all, a dehydrogenase enzyme, proline dehydrogenase, it was there, it is also here, it is converting it to pyrroline 5 carboxylate. This has not been shown over here, right, but it is there. So, it is first formed into pyrroline 5 carboxylate, right, which undergoes spontaneous conversion to this semialdehyde, right, and this semialdehyde is again acted upon by semialdehyde dehydrogenase to form glutamate, which is then finally transaminated to alpha ketoglutarate, right. So, the most important thing to remember over here is the fate of proline and presence of this compound glutamate semialdehyde. Okay, this one important, structure not important, names very important. Proline in protein is converted to its hydroxyl form. It has got a tendency to be hydroxylated by the enzyme prolyl hydroxylase, right. For example, proline is present in collagen and it is hydroxylated. So, there is presence of proline, there is also presence of hydroxyproline, right. So, it is hydroxylated, it is known as hydroxyproline. And for this hydroxylation, this prolyl oxidase, it needs multiple coenzymes. First of all, it needs molecular oxygen, right, then it needs vitamin C, iron and alpha ketoglutarate, right. And if there is excess degradation of this compound, it will again lead to production of hydroxyproline. So, hydroxyproline can be actually isolated from these proteins, right, but proline needs to be incorporated first and then it needs to be hydroxylated while it is inside the peptide chain, all right. A single proline generally is not hydroxylated by prolyl hydroxylase. Prolyl hydroxylase acts on the long peptide chain. So, since we read that this prolyl hydroxylase enzyme needs vitamin C to perform, right, and we also discussed that this hydroxyproline or formation of hydroxyproline is very important for the structure of collagen. The deficiency of vitamin C leads to collagen defect and the clinical feature is actually scurvy characterized with bleeding gums. There are multiple plethora of clinical features of scurvy and bleeding gum is one of them, right. So, basically what defect of collagen leads to? Collagen is a structural protein, so it leads to decreased strength of fibers, right. Now, excretion of hydroxyproline in urine is increased in infiltrating bone tumor and in diabetic patient due to enhanced rate of protein catabolism. I told you hydroxyproline is normally present in structural protein. So, whenever excess protein is being degraded, we can find hydroxyproline because hydroxyproline is directly a breakdown product of proteins and peptides, and whenever there is increased catabolism, we can find hydroxyproline in urine. Okay. Now, this disease imminoglycinuria, right. So, proline is an amino acid, right. Proline is an amino acid, it contains an amino group and likewise hydroxyproline is also an amino acid. So, this disease imminoglycinuria, as you have guessed by now, there is also glycine. So, whenever we are associating urea with a disease, it means excretion of these these amino acids in urine. So, why does it happen? It is due to defect in luminal reabsorption of these amino acids, these amino acids and glycine, that is proline, hydroxyproline and glycine. We have read glycinuria, all right, where we have also read a variant where there was defect in renal reabsorption of glycine. This is a combined transporter which is sodium potassium dependent, that is also ATP dependent active transporter, and it acts as a co-transporter. So, a combined co-transporter of sodium, potassium and these amino acids and glycine, which is defective in brush bordered cells of gastrointestinal tract as well as renal proximal convoluted tubule. So, the when it was supposed to be absorbed from gut, it is not absorbed. When it was supposed to be reabsorbed from urinary tubule, it is not reabsorbed, resulting in excretion of these amino acids and glycine in urine and stool. This is the disease of imminoglycinuria. You can have multiple choice questions, so be very mindful, right. We move to our next amino acid of discussion, that is arginine, highly basic semi-essential amino acid. If we consider the fate, it is glucogenic. What is so special about arginine? It has got a guanidinum group, right. These are special features regarding the chemistry of arginine. So, if we are discussing the fate of arginine, we already know, we have read urea cycle, and over there you know how arginine was produced as an intermediate of urea cycle, and the enzyme arginase was acting upon arginine to produce ornithine, which was again repeating the urea cycle. If you do not recall right now, it is high time you pause this video, have a look at urea cycle and then resume it, right. So, what happens? Ornithine then undergoes further reaction, right, in the urea cycle. If we are not concerned about urea cycle, we just read in synthesis reaction just a few while back that ornithine can be transaminated during synthesis of proline. It can be transaminated to semialdehyde, same semialdehyde, very important, and that semialdehyde is being converted to glutamate by a dehydrogenase and ultimately by transaminase to alpha ketoglutarate, right. And you already know from urea cycle disorder that hyperargininemia is an inborn of arginine metabolism due to defect of the enzyme arginase, all right. So, an important product of arginine metabolism is nitric oxide or NO. This enzyme or this product is, it is a gaseous product and it is synthesized by the enzyme nitric oxide synthase or NO synthase, often abbreviated as NOS, right. And the number of cofactors that this enzyme needs are many: NADPH, flavin mononucleotide or FMN, flavin adenine dinucleotide FAD, heme and tetrahydrobiopterin, right. This nitric oxide has got very short side effect, it exerts its action quickly and it is then destroyed. So, this is the reaction, that is where nitric oxide is synthesized. The arginine is converted to another atypical amino acid, that is citrulline, right, and oxygen is converted to nitric oxide, right. Why it is eNOS? I will tell you very soon. Okay. And these are the cofactors, right, oxygen, NADPH, FMN, FAD and heme. So, this nitric oxide synthase has got 3 isoenzymes. So, let us discuss them one by one. The first isoenzyme or NOS1 or neuronal variant is also known as nNOS. It is seen in central and peripheral neurons, right. So, since it is present in neurons or nerve, it acts on the nervous system. So, cerebellum as well as in neurons of gastrointestinal tract, right, nitrogenic neurons, right. It is a cytoplasmic enzyme and it is activated by calcium. All of these facts are multiple choice questions. Second variety, macrophage NOS or iNOS. Why it is called I? Because it is inducible, right. Same thing, iNOS, macrophage NOS, inducible NOS or NOS2. It is mainly seen in macrophage as a neutrophil and it also present in liver cells. So, why do we call it iNOS or inducible? Because it is induced by cytokines, right. Cytokines are chemical compounds that are mediators of multiple reaction, for example, inflammation. So, few examples of cytokine is interleukin 1, tumor necrosis factor. So, these are the cytokines or chemical compound that can induce this enzyme to form nitric oxide, right. Again, this is cytoplasmic enzyme. One key difference from first isoform is it does not it is not activated by calcium. So, calcium has got no effect on this enzyme, right. The third variety is endothelial, very important, eNOS, right. It is in endothelial cells, platelets, endocardium and myocardium, right. So, in these sites blood is constantly flowing, right. These tissues mentioned are constantly in touch with blood vessels, arteries, and nitric oxide is actually constantly relaxing blood vessels. Hence, it is also known as endothelium derived relaxing factor or EDRF. It is located in the plasma membrane and this variety, third isoform, is actually activated by calcium. There are some debates where many scientists have said that NO and EDRF are different, but multiple research papers have finally shown that there is no structural difference and action of NO and EDRF. So, for all practical purposes, these are considered to be one and the same. So, these are few roles of NO, right. So, nitric oxide, it's and how the drugs that help in production of nitric oxide, antagonize production of nitric oxide, can be used for therapeutic manipulation. For example, in chest pain we need angina, that is a disease where blood vessels are narrowed and due to lack of blood supply there occurs chest pain. It can be treated when we can devise a drug or medicine which can increase production of NO. For example, nitroglycerin or GTN or sorbitrate. Often we see chest pain, one small heart shaped tablet is put under the tongue, same mechanism using nitric oxide, right. It acts as a vasodilator, it prevents platelet aggregation, it regulates blood flow, it lowers blood pressure. So, NO antagonists actually markedly increases blood pressure. It functions as a messenger molecule and also it mediates bactericidal action of macrophages, the action of inos, right. During multiple infection, those cytokines, they induce this production of inos and thus kills the bacteria, right. Since it is a vasodilator, it has got role in erection of the penis because it dilates the vessels of corpus luteum, okay. Carpora cavernosa. And very important, this action of NO via CGMP, alright. This is again a discussion of signaling and messenger, but you should know, if you are studying nitric oxide, for your own interest you can go to the biosignaling chapter of your any textbook biochemistry and over there you can read how NO acts, right. So, finally, we look into the interrelationship of amino acids as we always do. So, we can see proline via formation of delta 1 pyrrolidine 5 carboxylate is converted to glutamate gamma semialdehyde, right. We already read that arginine via arginase is converted to ornithine and ornithine can be transaminated to form the same semialdehyde. That semialdehyde is ultimately converted to glutamate by a dehydrogenase enzyme. We have also read that this histidine by action of his multiple steps is converted to glutamate. We know glutamine, we have read during ammonia metabolism, can be converted to glutamate and ultimately by GDH reaction, glutamate dehydrogenase, it can form alpha keto glutarate. So, you can see so many of these amino acids are forming alpha keto glutarate, alright. So, all of these via glutamate is getting converted to alpha keto glutarate, okay. So, we are left with lysine. The lysine is also a basic amino acid. So, why we are discussing lysine separately? Because in spite of being an essential amino acid and basic, it is a ketogenic amino acid. So, its metabolic fate is actually different. We should know serial proteins are deficient in lysine and it does not participate in transamination reaction, right. So, if we consider lysine in protein, it can be present in multiple forms, that is lysine residues are often chemically treated, hydroxylysine, methyl lysine and acetyl lysine. So, these derivatives can be hydrolyzed to form free lysine, right. So, like hydroxyproline, hydroxylysine is also present in collagen. Now, when we are discussing methyl lysine, the methylation is done by the common methyl donor, that is S-adenosyl methionine. We already know that, right. And these when these proteins with methyl residues are degraded by proteolytic enzyme, it will release methyl lysines as well, okay. So, why do we need to know about methyl lysine? Because whenever because methyl lysine is actually a cofactor, I mean a precursor for carnitine synthesis, right. So, these are the steps by which carnitine is synthesized, okay. The intermediate enzymes and the intermediate products are not necessary for you to remember for any competitive exam, but in case of an exam like national common entrance exam for medical exam, there might be some image based questions where one of the intermediates might be removed and you will may be required to fill up using an image based answer. So, one of the 4 answers will be given and you need to choose the right one which fits the puzzle, right. So, but the most important thing for you to know is trimethyl lysine serves as a precursor, and what is the biological important role of carnitine? You have already read when we were learning beta oxidation and oxidation of fatty acid, that carnitine is basically involved in transport of fatty acid to the mitochondria, right. So, if we just look into the what are the steps, the first step is actually an oxidation step by a dioxygenase enzyme, followed by a splitting and reaction by aldolase. You already know aldolase always cleaves and it leads to formation of glycine residue is given off. Thereafter, by action of a dehydrogenase and another oxidation, we get carnitine, right. So, what are the functions of lysine? So, lysine and hydroxylysine are present as important product in or important residues in collagen and elastin, and they help in structural cross-linking of protein, right. Lysine has got an epsilon amino group. If you look at the structure of lysine, the amino group is present in the epsilon carbon, right, and it can form Schiff bases by linking with proteins, and this lysine is also found in high quantities in histones. Histidine is also found, right, and because these are basic amino acids, this is closely associated with nucleic acids. Finally, we need to know the ketogenic fate of lysine. We already discussed what is the fate of lysine, right. So, in this reaction, what we need to know is this intermediate alpha keto adipic acid. So, whenever we remember alpha keto adipic acid, which you have also learned when you are being taught about tryptophan metabolism, right, because tryptophan, the fate of tryptophan also lies in the same pathway. So, lysine and hydroxylysine actually forms alpha amino adipic acid, which is then transaminated to form alpha keto adipic acid. It loses one carbon dioxide decarboxylase enzyme to form glutaryl CoA, and once we get glutaryl CoA, there are three more intermediates. One is glutaconyl CoA, crotonyl CoA. You have read glutanyl, from glutaconyl CoA to crotonyl CoA during metabolism of branched chain amino acid, right. If you do not remember, you should revise your lessons properly. So, ultimately once we get glutaryl CoA, you already know that crotonyl CoA dissociates to form acetoacetyl CoA, which again further splits up to form two molecules of acetyl coenzyme A. So, if we look at the metabolic fate of the ketogenic amino acid, you can see tryptophan, right, as well as phenylalanine and tyrosine forms acetoacetyl CoA. Leucine, branch chain amino acid, was also forming acetoacetyl CoA, right, and isoleucine is also forming both acetyl CoA and succinyl CoA, right. So, we have magnified this thing. If this is for you to just recall what was taught before, right, so that you can connect all the dots over here, and finally you can also join all the things, all the amino acids to their common ketogenic fate, which again lies over here, which ends over here, right. And mind it, isoleucine, since it was both glucogenic and ketogenic, it forms succinyl CoA and acetyl CoA, which was already discussed during branch chain amino acid metabolism. And so, if you can actually recall, all of the amino acids have now been discussed at this point. You may think, okay, only one amino acid was left, so it is not basic, it is not related to this, but it is very important to know that aspargine, by the action of asparginase, is converted to aspartate, and you already know the action of AST, by which it can be converted to oxaloacetate, and thus it can also enter the final pathway of metabolism of carbon skeleton of amino acids. So, at this point you should be able to know and answer what are the sources and what is the metabolic fate of all amino acids that is shown in this diagram, all right. So, to conclude, this chapter we have discussed the metabolism of histidine, how histamine is formed, how histamine is degraded. We have discussed about metabolism of proline. We have discussed about metabolism of arginine, nitric oxide, role of nitric oxide. We have discussed the metabolism of lysine, what is the common metabolic fate, what is the common fate of glucogenic amino acid, what is the common fate of ketogenic amino acid, and all the amino acids are somehow connected in a common pathway. These are my references, and I thank you for your attention.